morning, and thank you, Jake. And it's a big one today. But first, before we get into the award, I would like to thank the subcommittee, the Redfield Award Subcommittee. So if any of you here are from the subcommittee, please stand up so we can recognize your hard work and clap for you. And we'll clap anyway, because even if there's no one here. Okay, so today is it is ASLO's Lifetime Achievement Award, and it is an honor and a privilege to introduce this year's Alfred C. Redfield Lifetime Achievement Award to Dr. John Smol for his leadership in the development and application of the field of paleolimnology. His work has established the use of lake sediment records and indicator species to identify water quality problems, monitoring ecosystem fluctuations, and to track changing climate. Over the course of his career, Dr. Smol has contributed to over 700 peer-reviewed publications, 23 books, and over 1,200 presentations around the world. He has mentored about 100 graduate students. Many have continued to work in the field of limnology. A nominator for this award wrote, and I quote, "He has singularly made the most significant contribution to the advancement of sediment-based lake research over the past 4 decades. In addition to his research, Dr. Smol has demonstrated exceptional leadership in the scientific community through long-standing service on editorial boards and engagement with policymakers. Dr. Smol is an active ASLO member, organizing meeting sessions, serving on ASLO committees, and being a long-standing associate editor at L&O Methods. Taken together, it is clear that Dr. Smol has been a lifetime advocate of the field of limnology, and his mark on the field will be visible far into the future. However, as John would quickly note, papers and awards, and I'm quoting again from one of the nomination letters, papers and awards are only the foundations. Scientists should do all they can to ensure that the implications of their findings are used to improve the environment for society. Thus, he and his lab have regularly gotten their message out in multiple media, and often to provincial and federal government politicians. In recognition of his exemplary career, Dr. Smol has been named a Fellow of the Royal Society of Canada in London and an Office of the Order of Canada. In 2004, he was also awarded Canada's top science honor, the NSERC Hertzberg Gold Medal. And here today, I'm happy to call on Pat to present the Redfield Lifetime Achievement Award to Dr. John Smol.
And I too am losing my voice. But on behalf of myself as president of ASLO, on behalf of the board and the hard work of the awards committee, I present this Lifetime Achievement Award to John Smol. Thank you. Thank you. The podium is yours.
Oh, thank you. Well, thank you very much for this this great honor, and I'm delighted to be here. I've been a member of ASLO since I was 21 years old, and I've been a consistent member since then. I'm not 21 anymore. It's always sort of been my home society, and it's a great honor to be here. What I'm going to try and do today is talk about history to some extent, partly a little bit of my history with a bunch of thank yous near the beginning, and then I'd like to talk about one recent project that we're doing, which I thought of. Perhaps this would be a good example where we start bringing a whole lot of different disciplines together, and that's what a lot of what paleolimnology has been doing over recent years. But first, let's go, again, ASLO has been thank you, the nominating committee, thank you to Shelley Arnott, who did the nomination, the letter writers, and everyone. I'm absolutely delighted to be here for receiving this award. I also like to acknowledge Canada. For the 40 million Canadian, no. I mean, when I was thinking of writing here, I thought about it. You know, a son of a war refugee and a political defector who met in the immigrant sections of Montreal somehow by chance. Of all the places I could have been born, what plain blind luck that I was born in Canada. I was born in a country where I can call the Prime Minister a fool and go to bed at night and sleep fine. Also a country with thousands and thousands of lakes. So I really do appreciate, I'd have to keep reminding myself of how lucky I've actually been in many respects in my life. Also like to thank NSERC, they're our funding agency. NSERC has stood by me from day one since I was a graduate student. It's our federal funding agency, as many of you know. It stood by me when I was the most basic or foundational type of research of taxonomy and basic ecology, all the way to do heavily applied research. And it really has allowed me to run a lab going in directions which we felt were important. Of course, I've been at Queen's University for my entire career. We'll be reaching the 40-year mark. I was a very young professor, if you're calculating my age, but reaching the 40-year mark with excellent colleagues, excellent limnologists, Shelley Arnott, of course, who many of you know, Diane Oriol, a former ASLO winner in Spain, as well as soon I'll be talking about also members of my lab, the Paleoecological Environmental Assessment Lab. But it is a bit of, as I say, I tend to be a lake historian, I guess, in some ways, and I tend to look backwards. And of course, we have to acknowledge where we started. Sadly, Jacob Kalf died a few weeks ago. But at McGill University, I did my undergraduate at McGill. He was my limnology professor, first instilled my interest in limnology. I had also the good fortune of my ecology professor to be Rob Peters, who died tragically quite young, but definitely key people in my life. I did a master's at Brock University with Mike Dickman, and then I really got into paleolimnology when I actually did my PhD at Queen's. And I did that with S.R. Brown, or Ted Brown. That's a picture from, we believe, about 1953. Ted's on the right, and Ed Devey on the left. And this was early paleolimnology, but they were really the pioneers doing the sort of the back work to make this field what it is today. At that time, paleolimnology was seen as much as my early career, was seen very much as a side issue, sort of something in geology or geography. We'd all go, about 5 of us kept going to ASLO meetings, but we'd often be on Friday afternoon, and the 5 of us would talk to each other. We had projectionists then. There'd be a bored projectionist sort of sleeping in the back, and we'd talk to each other. This has changed totally. We have paleolimnology. We don't have really paleolimnology sessions. It's integrated into limnology, and that way, I think that's a very major thing to do. Following my PhD, I got a postdoc with the Geological Survey of Canada to work in the Arctic, and my main mentor then was Wes Blake. Wes Blake I met at a conference, one of my first conferences, and he was sort of interested. He was a glaciologist, was kind of interested what I could do, or he thought I could do, and invited me to go to the high Arctic when I was quite young on his expedition. That started 40 years of Arctic work, and I'm just celebrating 40 years of working in the high Arctic. There's a picture of Wes. On the left is a picture of Wes when I took in 1983, the first time I was there. The next picture is on the same rocks in 2009. He took me up the first time, and then once he retired, we took him up several times. It's pretty well essentially the same rock. Could be the same pants, but now that I look at it, but anyways, of course, this became my interest and my still ongoing interest in polar regions, which I had the pleasure of recently writing, which is part memoir, part textbook, this 13-chapter book on lakes in the Anthropocene, and it's all the different ways that we could actually do work in the Arctic. I was never a student or postdoc of David Schindler, but he was always an important influence in my life. I heard about him when I was an undergraduate, when he was starting to do many of those ELA experiments. We became friends. He was always a role model and someone that you can do intense science, and he did intense science, but would take that science to the public, take it to politicians. And he made the time, and he really instilled in me that's critically important. And he was an important influence in my life in that respects. And also Bob Wetzel. I met Bob Wetzel at my first ASLO meeting when I was, by my calculation, 23. He came to my talk. I couldn't. He was like he had. I mean, he was just like someone I knew of from the textbooks, of course. He came up to me, talked to me like I was a scientist. Here I was just a master's student at the time, and I got to know him much better over the years. Sadly, of course, he also died quite early. I think he was 68. His textbook was amazing to me, at least. It's stunning when you look back. He wrote the first edition of his textbook when he was in his 30s. And he kept writing different versions, writing the last version in just shortly before he died, 2003 it was published, or 2001, and again, 2001, sorry. And when you realize how he did those early textbooks with paper reprints, and he kept his references on index cards, it was quite remarkable. We had the honor of being asked to do a new version of Wetzel's textbook. We have to keep his name there because he was almost like a silent author on many of the chapters, but it took 59 authors and 2 editors to do what one man did. And when we got the comments back from authors, they just kept saying, "I don't know how one man did this." And we had 2 or 3 authors on almost all of these chapters, and it was really quite remarkable. So these are people who have definitely been important in my life. The big thank you, though, goes, of course, to my lab, the Paleoecological Environmental Assessment and Research Lab, or PEARL as we call it. Over 100 graduate students have passed through my side of the lab, and Brian Cumming, who I mentioned a minute, has many more, not many more, but more, and postdocs. If people say, what is my biggest honor, it is the students and the postdocs that have gone through my lab. I'm incredibly proud of, and many of them here too, how proud I am of what they've managed to accomplish in their own areas of primarily paleolimnology and limnology in general. About as close to 10 years ago, I gave a keynote, and I didn't realize this, but they had done a tree of my students up until that time. That's what's sitting on the left. But there's 20 or 30. There's a lot more branches now. There's about 20 or 30 more people, and I'm not. You get a Lifetime Achievement Award, but I'm not really finished yet. I just took on 3 new PhD students and 2 new master's students, so you're going to be stuck with me for a while. But I do want to begin by saying, and go to some other acknowledgments, getting awards is a bit like, to use a hypothetical example, like the Toronto Maple Leafs win the Stanley Cup. It's hypothetical. And only the coach goes out there to hold the trophy. I mean, anything I'm going to be talking about here, or what I've done, has been a remarkable group of people in my lab who have worked with me and continue to work with me and are my closest friends. So when it comes to, I can't acknowledge all the hundreds of people. I'm going to acknowledge about 4 or 5 here that have been with me for a long time. One of them was my first PhD graduate, Brian Cumming, who many of you know. He's now, he went to University of Minnesota for a postdoc, then got hired back at Queen's, which was quite rare. And he's the co-director of the PEARL Lab. He's also now head of my department. So for the early career researchers out there, it's a good little lesson here. Be nice to people on the way up. You may well see them again on the way up. He assigns my teaching, my committee work. So anyway, there's a good life lesson that perhaps we have there. And again, there's so many colleagues, but 2 that I feel I have to mention have been long-term research scientists in my group, Neil Micheludi and Kathleen Ruland. I think many of certainly the diatomists and the paleolimnologists know those names. They've been with me for a long time. Again, outstanding scientists who are in Kingston for family reasons. Their spouses are in the university, and so they're on. And I love field work, loved field work, getting a little harder, arthritis in the wrist and things, but I'm not overly practical when it comes to fixing engines or sewing up tents after a polar bear tore them apart, but I've been blessed with two excellent people who have worked with me. John Glue, again, many people may know, sadly died suddenly in 2019 unexpectedly. Built most of the equipment, the specialized equipment we use. That equipment is now used on all 7 continents. And continuing on now with Chris Grooms, my lab manager, technician, and also, again, a close friend. I have other colleagues outside Queen's University, one of which is Jules Blais. Jules Blais is actually my half-brother. We share a mother. But he's also a paleolimnologist, also a former Lindemann Award winner, Rigler Award winner. But he works at University of Ottawa. No one would guess we're brothers. He's tall, thin, blonde, has a French-Canadian name, good-looking. But yeah, we share a mother. We share a mother, so we are related. But he's much more chemical than I, and I do the more ecological stuff. So he's a co-author on many of the, going to be a co-author on some of the examples I show here. Andrew Patterson on the Ontario Ministry of the Environment, also a former student, works very closely with many of my students in the applied field, and he's still on as I say, on the front edge of applied science with us. So those are just a few that I feel I would like to. I'd like to keep going. There's many, many more, but I think that's all I have time for. I'm going to give you an example here of some of the things we are doing with paleolimnology, and these are the important policy questions we typically have when it comes to environmental work. What were pre-disturbance conditions? I cut my teeth on acid rain doing paleolimnology in my 20s. That was a classic question. What was the acidity of these lakes in the 1850s? People were claiming, yeah, the lakes are acidic, but they've always been acidic. How do you show they acidified? You need to find out what the pH values were in the 1800s. Sorensen only invented pH in 1909. We could use our methodology to show that. What is the range of natural variability? Climate change is a good example of that. Yeah, sure, you can look at the readings, but maybe that's just some part of natural variability. Have conditions changed? How much? How fast? When? Why? Each of these questions has a time or a temporal component associated with it. And that's one of the biggest challenges we have in ecological environmental research, the question of time frames. I did this analysis back in 2019, and I asked a simple question. I went to the International Journal Environmental Monitoring Assessment, and I asked in the previous year, what is the time frames of the aquatic studies in this monitoring version? And as you can see, about 2/3 are less than 1 year in duration. Over 90% are less than 3 years in duration. That's not to say 3 years or less is not enough time to do serious research. Of course it is. But it's difficult to answer some of those questions I posed in the previous slide using that type of analysis. So this is what I really want to talk about, the issue of timescales and what we can get teasing apart the past, leveraging the past. History is important. History warns us of our failures. History tells us what worked. To use Ed Devey, I showed a slide of Ed Devey earlier. He wrote a paper once called Coaxing History to Conduct Experiments. If you do it carefully, you can get history to actually do a quasi-experimental type approach. And the overall approach is remarkably straightforward. Lakes and oceans, of course, as paleoceanography uses very similar techniques. Lakes slowly fill with sediment 24 hours a day, 365 days of the year. There's material coming from outside the lake, things like pollen grains, contaminants. Also within the lake to like algae, chironomids, all sorts of different things, chemical precipitates, and that makes a depth time profile at the bottom of the lake. Our job as paleolimnologists is to reconstruct that history and interpret it in a way that's meaningful to other scientists, policymakers, and the public at large. Now, our lab over the last 40 years has done a large number of different types of projects. Here's just a few of them, some of them going from very foundational or fundamental, like long-term lake development, acid rain, eutrophication, well, the list goes on and on and on. There's many, many things that you can actually do with that. And on top of that, of course, climate change. And back in a paper in 2010, I called it the new threat multiplier. Almost every issue I deal with is made worse in a warming planet. So now we have the complexities of dealing with multiple stressors. And if I do look back at my career, I went everywhere from being the most foundational. My PhD thesis was how do lakes change since the last ice age? What could be more esoteric than that? Then suddenly in my 20s, very early in my 20s, acid rain became an issue, and many of us realized we can use this technology or these methodologies to reconstruct acid rain situation scenarios. So in a blink of a week or so, I became from being a sort of a basic versus an applied scientist. And I think what I really learned there, the main difference between a basic and applied science is a matter of time. And I'm the first to acknowledge that I can't do any of the applied stuff I'm doing without that foundational work that were done before me or were done by our labs. The overall approaches are fairly straightforward. You get the head of your department to take your sediment core because it's get wet and messy. And these are the glue cores. They've been used around the world. This is especially good for the recent sediments about the last 200 years. They don't mix the profile. We can section them at fine intervals. And there's a whole spectrum of what I might refer to as traditional paleolimnological markers, things like pollen grains, contaminants, diatoms, cladocera, insect fossils, but there's a whole spectrum of other indicators. And then, of course, we have to set the time profile. If you're going back in time, you use C14, radiocarbon. If you're in the most recent, and much of my work is the last 200 years, we're actually using lead-210, many of you are familiar with it, natural occurring isotope of 22.5 years, 22.25 year half-life. So you can get a sediment core, and instead of just saying this is 20 centimeters or 15 centimeters, you can actually put dates around it. And here in this sediment core, this is one taken in Ontario, to use a Canadian example. There's 1867 when Canada became a country. There's the last time the Toronto Maple Leafs won the Stanley Cup. There's a lot of sediment that has accumulated since then. But that's the overall approach. And of course, there's many new indicators happening. So I'm here at ASLO, and I remember there's an O in ASLO, so when I was thinking of examples, I tried to think of something that included oceanography. I'm basically a limnologist, and what I thought I would look at is give one recent example done by many people in my lab, most notably Matt Duda, a large part of his PhD thesis recently, and as well as Jules and other people. And I'm going to look at bringing paleolimnology into conservation biology. And we've been doing sort of conservation biology stuff for a long time when we reconstruct habitats. We've been doing this forever. Well, not forever, but, some years. We do things like we reconstruct deep water oxygen levels for lake trout habitat. We reconstruct changes in food sources, changes in pH. So we've been doing that for a long time. But what we've done less so is actually looking at trends of individual species of interest and blending the two together. And that's what I think conservation biologists really like. And why seabirds? There's a lot of interest in seabirds. Seabird populations have declined by around 70% since 1950. And we only know for like 1 out of 5 species of any types of data, so seabirds appear to be in big trouble. Seabirds also are greatly, they're very important, as you all know, as biovectors in moving nutrients and contaminants around. These are remarkable numbers, the annual seabird input inland, how much nitrogen and phosphorus coming in. It's comparable or even higher than global fisheries. So seabirds are a key component of these ecosystems. So we have this schematic that I showed you over there, and since seabirds either fly over the lakes, or more likely what I'm going to look at is colonies that drain into lakes, because they change the limnology so much, and by knowing the limnology we can find out what those limnological changes are, we can go back and reconstruct seabird populations going back in time. And I'll give you one example, a recent example. Leach's storm petrel, one of the most common seabirds over the ocean. It's recently been classified as vulnerable. And it's an ocean piscivorous fish. It eats primarily lanternfish, and it nests on islands typically, and it's a burrower. There's a young chick there. And it's believed that there's a it's an important seabird, one of the most common seabirds there are, indicating over 30% decline just from 1977. Here's the life cycle. So there's a collection phase. They go and they feed out in the ocean. They collect phosphorus and nitrogen and contaminants, feeding primarily lanternfish, high up on the food chain. They fly over to their colonies. They nest there, rest there, defecate there, die there. All those nutrients are making their ways into the lakes. I'm going to take you off the coast of Newfoundland, Baccalieu Island. It's the largest colony in the world with 4 million storm petrels. So we went to the largest colony in the world, and And on these, those little dots that I'm putting on there, those are lakes that are on that island, Bacalou Island, and there's one control lake which is away from seabirds over there to the left. Why are we going to Bacalou Island? People were very nervous about Bacalou Island. It's the largest colony in the world, and they had two data. Now, ornithological data is very hard to get. They had 2 data points from the Canadian government, and they saw this trend. Now, that got them scared. We got the largest colony in the world, and there it goes. But it's 2 points. I said, maybe that's the line. But how do you do that? You can't go back in time and do that. Well, you can go back. It's a 40% decline. You can go back with paleolimnology. And here's a map of Bacalou Island. There's high abundances of the birds, moderate, low. So we have a gradient. So we had 4 ponds on Bacalou Island and 1 reference pond. And they had varying amounts of birds. The ornithologist could tell us how many birds were there. And we had 1 reference pond away from the birds. No animals were hurt in the study. These were dead birds that we found. The ornithologist gave them to us. We studied the bird itself, the feces, what the chemistry of the feces is, what the chemistry of the bird is. Not very surprising given their diet. They're very acidic feces, very high in nitrogen and phosphorus, high in cadmium and zinc. Because they're piscivorous, not surprisingly, they're very high in N15 isotopes. We're also looking at sterols and stanols. That's part of Jules Blair work at University of Ottawa. They have a different sterol and stanol formula. You can't do paleolimnology without the limnology. We first do the limnology of the ponds, and sure enough, there's the range of phosphorus. The reference pond's at 12. We've got, as you go higher and higher, bird print. Look at Brister Pond, 146. Look at Lunan, 138. Clearly, they're bringing in nutrients. These are not typical Newfoundland ponds. What about things like cadmium or zinc, other things? There's the line for toxic for aquatic life. There's the reference pond. As you can see, they are bringing in a whole spectrum of cocktail of elements that are very closely related to their diet. So what do? Well, we make some predictions based on the limnology. If we're going to reconstruct birds, we should see an increase in N15 because they're high up on the food chain. Increase in nutrients, we should see acidification, and we should be able to track that with diatoms and chironomids. We're going to see an overall increase in primary productivity. We can do that with fossil pigments. We should see an increase in specific metals that are in their diet, cadmium and zinc. And because they're very high in cholesterol, something I know a little bit about, very high in cholesterol, we can also track it with ratios of different sterols and stanols and sediment. And we looked on about 5 other proxies. I don't have time to even bring those up, but there's about 10 proxies that we looked at. This is Lunan Pond, one of the very eutrophic high impact sites. This core went back 1,400 years. We have longer cores I'll show you in a minute. But when we did the first comparison, going back 1,400 years. What we have here, remember, and we have many proxies. Here I'm only just showing diatoms and chlorophyllae. What we see is, if you focus on that part of the slide, those are acid diatoms. And because they acidify the lake, we said those are the most likely spots where there were bird colonies in the past. Especially at the top. It's a less acidic species down at 1,400 years. That also matches the chlorophyllae. It also matches all the other proxies. I'll show you a summary slide in a minute. But that is the 2 periods of seabird. And we do actually catch the decline over the last 20 years that the ornithologists have caught. So we actually do see those declines. Here's another one, 2 more high impact ponds. Again, the same type of stories. We see this enormous increase about early 1800s, this big increase in the early 1800s, and we still see, in fact, the decline at the top. It matches the chlorophyllae, and it matches 8 other proxies, which I don't have time to show you. What about the moderates? One site had only a few birds. It again matches perfectly. There you can see their acidic species are increasing over, and the chlorophyllae increasing 200 years ago. But again, and the decline over the last 20 years, just what the ornithologist said. Reference pond has no acidic species, no changes. Chlorophyllae is a straight line through the process. If you put here's more proxies, I put in more in this one, they're all sort of matching. And it looks like over the last 1,700 years, that's the trends in storm petrels. And the green represents the 2 distinct peaks, especially at the front end of the last 200 years. You put all of them into the Z-scores. Here's a 1,700-year history of storm petrels on Bacalou Island. So, now, there's some context here. we do see the decline. Everyone, I think, sees that decline at the end here. But is this what's stable? What's natural? This figure blew the ornithologists away. They were more or less expecting to see a straight line and a decline at the last 20 or 30 years. This changes the view quite significantly. It means these birds have been fluctuating remarkably in population, and in fact, for this region, have increased during the Anthropocene period, during the period of warming. We push this back to going back to the Ice Age, 12,000 years. We did more cores going back to the Ice Age. I can't get much into this, but certainly during the warmer periods, it's published in Global Change Biology if you want the paper, but during the warmer periods on Baccalieu Island, the birds did better. Okay, that's one lake. We then thought n equals 1. We can't say too much. Let's go to another large colony. The second largest colony doesn't have a lake around it, so we went to the third largest colony. Third largest colony was in French territory on the islands of Saint-Pierre and Miquelon, which are just about 270 kilometers away from Baccalieu Island. There's Matt Duda. We finally got permission to go to Saint-Pierre and Miquelon and look at the third largest colony. Now, what's about the third largest colony? Here's that's Grand Colomiers. So I just showed you. We were just, if you know your chemistry, we were just over here. Now we're going to go down to just off of Newfoundland is this last French territory there, and it's a potentially stable colony. Why do we call it stable? Well, the ornithologist, those are the lakes and ponds we're going to look at just like that. Ornithologists had 4 data points going back to the 1990s, and they showed that it was more or less a stable colony. With similar methods, the colony appears to be stable. So the ornithologist said, why is this colony stable and why is Baccalieu decreasing? But by now they're saying, well, it's decreasing from this enormous high that started 200 years ago. What if we extend the temporal scale? I won't get into all the details, but I'm going to use the same proxies. We had, again, about 10 proxies. I'm going to show 6 of them here. This is the proxy going back since the ponds formed 6,000 years ago. We do see, so this is supposedly a stable population. Now, some interesting things here. I mean, it was sort of more or less, we do see, if we do a zeroing in on the last 40 years, which I won't show you here, but it's in the paper, yeah, it's quite a stable population. But it's only 16% of what it was pre-1816. Why 1816? That is when the French, remember the Napoleonic Wars are coming to an end. This is the last territory they have in the New World or in North America is these 2 tiny islands, Saint-Pierre and Miquelon. They sent all sorts of settlers here. And you can see that we have a very different scenario. The current colony is only 16% of what it was pre-1816. So is this a healthy, stable population? Again, context is key. Why did it suddenly crash? This one we have a pretty good reason. Storm petrels, they were even used as candles on these islands. There were so many of them at the time, but they were eaten. They were sometimes eaten, but they were apparently tasted terrible. They mainly used as candles. The island, there is evidence of a shipwreck with rats, and right around this time, and they talk about rats running through the island in people's diaries and other things happening. But here we have a completely opposite trend to Baccalieu Island. Not just since 1800s. Remember, since 1800s, Baccalieu Island went way up. The third largest colony, only 270 kilometers away, crashes. And in fact, if you go back, Saint-Pierre and Miquelon only goes back 6,000 years, but they appear to be all in reverse. When one is doing better, the other one is doing worse. Now, when we showed this to ocean modelers in Newfoundland, they said, exactly. That's exactly what we were thinking. And of course, I didn't understand. They use a whole lot of Greek letters. I didn't understand much of what they're talking about. But they said, they're only 270 kilometers away, but they're very different ocean environments, different currents. The pycnocline, they're talking about the Labrador current and other currents. They say that's exactly when one is warmer, the other is colder. When it's colder, the other is warmer. And that was actually, they were very excited about these data, saying that's exactly what our models were suggesting was happening in the past. So they were very happy about it. So we actually have many, many drivers of what's happening, local factors such as human impacts, regional drivers such as climate change. And again, when you look at this, Baccalieu Island, those are the 2 data points. Based on the available census data, the petrel population declined by 30%. But we do see the 30% decline, but context is key. I mean, that is the decline within a perspective of the last last two millennia. Is this a stable population? I don't think so. But it is based on the ornithological data. Again, context is key. And of course, we've done this with many other birds. If you're interested, just let me know. And if you have other sites, basically we need a lot of poop and a draining into a lake or an ecosystem. So just looking back, I'm at minus 4 seconds. Okay, but it's my last slide. Lessons from the past. I mean, certainly if I learned anything by doing this, the recurring pattern of unintended consequences. My 40-year career, there's always things we're not really thinking about. Things sound great at the time. I mean, my Arctic book, I have a subheading, it sounded like a good idea at the time, and all the other things that happened since then. We also tend, if anything, to be overly optimistic. Things are generally worse and more complicated than we initially imagined. And I'm not suggesting you Google John Smol, but don't read everything you read. Eco-fascist and everything. Most people see me. I've been an optimist when it comes to environmental work. We didn't really appreciate all the issues that are there, and we're rapidly running out of time. The world, I'm speaking to the converted here, but the world's gone insane. I mean, even the vocabulary is changing. It wasn't too long ago that someone studied something for 30, 40 years, they'd be called an expert. Now they're called an elite. It wasn't too long ago that we called things environmental legislation. Now it's quite easy to say it's just red tape. It wasn't too long ago when we were talking with industries and they were talking about futures, we used to say they were pulling numbers out of their asses. And now we say aspirational targets. I mean, these are, who comes up with these words? Aspirational targets, pulling numbers out of our asses. That's what it is. But we have to, I'm talking to the converted here, we can't give up. If history teaches anything, things are worse than they are. And to paraphrase Emerson, nature is slow to pardon any of our mistakes. So thank you very much for this great honor, and I'm very pleased to be here. So thank you. I'll take them, but I was at minus 17 seconds.
If there's maybe one quick question from the audience, otherwise we can chat at lunch.
Yeah, I'll be here all the time. Oh, thank you very much.
Thanks, John.